Good morning, students. Today we will be discussing amylase production. So, first let's discuss that what is amylase. Amylase is enzyme, right, which is usually used to hydrolyze starch. So, amylase enzyme. So we are discussing your commercial production of amylase, and we know that commercial production of amylase. So before I move on to the into the amylase, we first need to know that have you heard about digesting or your protozyme or your orizyme? So these are basically some of the trade name of digestive enzymes. And these digestive enzymes are basically produced by your microorganisms, and they are basically having fungal amylase. So amylase is used in pharmaceutical industry. Amylase is also used in your textile industry. So let's talk about the learning outcome of this video. That means we will be focusing on the microorganisms which are being used in amylase production, the substrates which are being used in the amylase production, and the different kinds of fermentation process which will bring about the industrial production of amylase. So these are some of the focusing points we will be discussing today. Now, as we know that the bacterium which is being used in amylase is your Bacillus subtilis. So this is the bacterium which is being used in amylase, and there are many molds also which is being used in amylase production. So we will be discussing it one by one. That is your Rhizopus, dilemma, Mucor, Roxy, Aspergillus, Niger, Aspergillus oryzae. So these are some of the molds which is being used in the amylase production. So next we will move on to the microbial production of amylase. So let's start with the microbial production of amylase. Now as we move on, we know that increasing amount of amylase are being produced from the molds, which may yield primarily one kind of amylase or mixture of amylase. So mold can produce one kind of amylase, or it may cause mixture of amylase. Okay, so that is one point. Next point is that in this method, the bran are thoroughly steamed. So basically the substrate is your bran. And this particular bran is being steamed. So it is being steamed, and after steaming for 1 to 2 hours, it make this. When we steam it, we basically do why we steam it. We steam because it makes your starch soluble. So it makes your starch soluble. Okay. So once you steam it, and then 1 to 2 hours, to make starch soluble and to make the microorganism, to kill the microorganism, then the bran is cooled. It is being cooled to 25 to 30 degrees C. So 25 to 30 degree C, it is being cooled. Then the cool material is inoculated. So the cool material is inoculated with the starter culture. Starter culture. Now when we talk of your starter culture, we should know that what starter culture we usually take, and the starter culture over here usually Aspergillus oryzae. Aspergillus oryzae is the starter culture. So the inoculated mass, so what is happening? So next point, the inoculated mass is well mixed and spread uniformly in trays. So they are being uniformly mixed and spread onto that of the trays. So they are being mixed and spreaded to the trays. And then after spreading to the trays, then the fermentation temperature is maintained. So the fermentation is started, and the fermentation occurs at 30 degrees C. Now, high humidity and sufficient ventilation are necessary for the oxygen supply and to eliminate carbon dioxide. So, you know that your high humidity and ventilation is very essential, because that will give you oxygen supply and remove carbon dioxide, right? And then the mold develops rapidly. So, your mold develops rapidly, and this mold develops rapidly within 46 to 48 hours. So they basically develop within 46 to 48 hours, and then during this period, mold mass, the maximum produces a maximum desirable enzymes are produced, and the product may be dried to prevent bacterial action. So now the product is basically the product, that is your amylase, is basically dried. So why it is being dried? It is being dried to prevent bacterial action, and ultimately then it is being extracted by 1 or 2 volumes of water, 1 or 2 volumes of water, and then it is being preserved. Then it is being preserved. So this is the basic mechanism of your amylase production. Now, if it is for the medicinal purpose, so if it is for the medicinal purpose, if it is for the medicinal purpose, then you know that 70% ethanol is added. And why it is added? Because that will added for saturation. So this will basically precipitate down the amylase, and it is being dehydrated to produce your powder. So it can be produced in powdered form, okay? And this powder is basically your whitish yellow in color. So it is basically whitish yellow in color, right? And it is, if it is used for other purpose, about 20% NaCl is added and can be preserved using preservatives such as phenol or thymol. So if it is being used in other purpose, then you can also add 20% NaCl, and that is being preserved, and preservatives are basically phenol and thymol, okay? So this is the overall production of your amylase, the flow chart of your amylase production, right? So next we will move on. So next we will move on to your preparation of inoculum. So let's move on to preparation of inoculum. Now, when we talk of preparation of inoculum, Aspergillus oryzae is cultured in your agar slant, and it is used to inoculate sterile, moist bran. So, Aspergillus oryzae is actually grown in agar's land, and then it is being used to inoculate sterile bran. Now, what is happening? Adequate sporulation is required. So, this oryzae need to sporulate. Okay. Why? Because this is very important that if they undergo spore suspension, addition of spore suspension will result in huge amount of growth. Okay. So the thin film, the mold is used to inoculate, sterilize and pull bran. The mold is grown in specially designed shallow trays equipped with perforated covers. So if I draw the trays, so these are the shallow trays which is having your perforated covers. Okay. So in this particular tray, a thin film of paper or cotton cloth is placed on the top to prevent contamination and to absorb moisture. So once you lay down the moist bran, once you add the microorganism, you keep a cloth. And what will happen to this cloth? This particular cloth will basically absorb moisture and prevent contamination. Okay. So the trays containing the mother culture are placed in racks in special incubator room. So suppose there is one rack and the trays are basically being kept onto the rack. You will be coming to know that at what distance they are being kept, how they are kept, etc. So that is your incubator room, and the temperature is basically maintained at 78 degrees C with forced air circulation and by heating and cooling coils. So after the incubation period, till the spore formation, the mold spore preparation is ready. So the mold spore, once it is being ready, then we have to inoculate onto that of your bran. Okay. So next day we will be starting with the preparation and inoculation of the substrate.